So hi, I'm Tom Plunkett, and I'm going to give a brief presentation on an award we won last year working with the National Cancer Institute on analyzing cancer-genome relationships. And as the other speakers said, 5 minutes is really short, so please feel free to come to the poster session. I'll talk more about this. A little bit about me. I lead Oracle's big data practice for the federal government. I'm going to kind of skip through this, but basically I've been working with Hadoop for about 4 years. Also, I'm not going to talk about Oracle products in this presentation, but if you're interested in learning about Oracle products and what Oracle's doing in big data, there's a book that just came out that I'm an author of, the Oracle Big Data Handbook. And Doug Cutting had some nice comments to make about it. So let's talk about what I really came here to talk about, which is the project we did with the National Cancer Institute's Frederick National Lab for Cancer Research, which is based outside of Washington, D.C. Basically what we did was we used Apache Hadoop and R to perform analysis and examine relationships between different cancer types and different genomes. So why is that important? Well, in case you didn't realize, personalized medicine is rapidly approaching. A few years ago, it was extremely expensive to perform gene sequencing. Today, it's rapidly approaching under $1,000. So this is going to be the future. It's going to be what we're going to be focusing on. And unfortunately, there's been so much research on all these different gene types that you need some way to be able to, for a scientist or a medical professional, to be able to easily understand and comprehend the research that's been performed relating to these different diseases and the individual genes. So that's what we were looking at. We were looking at a way in which we could apply and understand this information, and we wanted to do that using, the new big data technologies to make it affordable. Go back one. Okay, so we looked at several different data sources we were dealing with. For instance, PubMed has a database of a massive number of different, I think over 22 million documents relating to this area. We also needed to use the Thesaurus, the Cancer-Genome Atlas, because obviously the documents in the PubMed database use all sorts of different terms to refer to diseases, in different languages, you name it. In terms of what the actual problem was we were facing this is how one of our research scientists described it. Basically what we're looking at is relationships from genes to genes, from genes to the drugs, and genes to the different diseases. Now, how do we interpret that? Well, you can interpret that as actually just looking at for relationships and making them easy to visualize. And here's how we actually converted it into, how would you give it as a requirement in software code? So here's what we came up with, and I got several different graphs that I'm going to show, and I've got these graphs on the poster outside, so we can talk about them in more detail if you have specific questions. But basically, on the left-hand side, I have different diseases listed, like for instance, NSCLC is a type of lung cancer. On the upper right hand, on the top of the graph, I've got the different genes, and so you can see, for instance, if you look at the column for NSCLC. Is that the pointer? Yeah, right there. All right, so you can see there's a lot of different genes that are related to this particular type of lung cancer. On the other hand, you've got some diseases like this type of melanoma that only has a single gene that's associated with it. And the size of the dots indicates the amount of research that's being done. So for instance, this particular gene disease interaction has a lot of research. On the other hand, a smaller dot like this has very little research. And then no dot means there's no research whatsoever. Now, we also looked at other ways of visualizing this information. For instance, here is the same information, but graphed differently. So for instance, over here, that's the same type of lung cancer, NSCLC, and you can see all of the different genes that are related to it. On the other hand, you've got some outliers that have very little relationship. And so this is another way of looking at it. For instance, outliers can be important because that can give you an idea of where you need to invest more for research. And thank you very much.